Today's topic is about 8.3, which is all about photosynthesis. But before we dive into that, let's review some of what we learned in 8.2, which had to do with the many steps of respiration. So, as previously discussed, respiration, instead of being one process, is actually really 4 different reactions together. And that respiration is about the breakdown, the catabolic reaction of breaking down glucose into ATP energy. And that the first step of that catabolic reaction is glycolysis, which produces 2 molecules of pyruvate, and that is occurring anaerobically in the cytoplasm. And then that pyruvate enters the link reaction in the matrix of the mitochondria, and pyruvate is there converted into acetyl-CoA. Once we're in the matrix and we have that acetyl-CoA from the link reaction, now this molecule can enter the Krebs cycle, which is again occurring within the matrix of the mitochondria. And the main point of the Krebs cycle is the creation of these electron carriers, NADH and FADH2. And so there were a number of important reactions that were happening within the Krebs cycle. We have the reduction reactions, which are creating these electron carriers. We also have decarboxylation events, where CO2 is being removed from these intermediaries. And that, remember, our number of carbons goes 6, 5, and then 4, right? Because I lose a carbon dioxide here. So then this would be 5 carbons. I lose another carbon dioxide there. That would be 4. So the rest are 4 until it regenerates again. And then you go back to 6 carbons. But again, the main point of the Krebs cycle is the creation of these electron carriers, because they are going to go off to the electron transport chain, which is itself occurring within the inner membrane of the mitochondria, also known as the cristae. And so once these electron carriers reach the electron transport chain, they are then being oxidized. And so the main point of this is the creation of a proton gradient, and you're going to use the power of chemiosmosis to power ATP synthase to make a ton of ATP. And then, so just a reminder that in the electron transport chain, oxygen is the final electron acceptor here from complex 4, which is creating that water byproduct. So that's what we covered last time in respiration. And so today we're going to focus on photosynthesis, which we know from both topic 2.9 and also probably the 3rd grade is all about producers using the light of energy to somehow produce glucose. And we also know from 1.2 that within plants, this photosynthesis occurs within a special organelle called the chloroplast. So like respiration, photosynthesis does not just happen in one step. There are many steps, but it is broken into 2 major phases: the light-dependent reactions, which obviously depend on light, and then the light-independent reactions, which are also known as the Calvin cycle. So what we're going to see is that photosynthesis in many ways is very, very similar, but kind of a reverse of aerobic respiration. And so like respiration, we're going to start with the structure of the chloroplast itself. So remember, both the chloroplast and the mitochondria are descendant from prokaryotes. So they're going to have structures in common with each other and with those prokaryotes. So the chloroplast, like its cousins, has both an outer membrane and an inner membrane. But it also has structures that are going to be similar to the mitochondria but unique to the chloroplast itself. So mitochondria had cristae, right, that crisscrossed across the width of the mitochondria. In chloroplasts, they have something different. They have a thylakoid, which are these stacked structures that are in the center. And within the thylakoid, there's open space in them called the thylakoid lumen. So the chloroplast also has empty space, which is called the stroma. So now it is no longer the matrix, it's the stroma in the chloroplast. And because the chloroplast is a descendant of the prokaryotes, it's also going to have those 70S ribosomes. It's also going to have a single circular chromosome. So chloroplasts are different though, because they are going to do this process called photosynthesis. And as I said, there's two portions to photosynthesis. The light-dependent reactions, which are happening in the membrane of the thylakoid, and then you also have light-independent reactions, also known as the Calvin cycle, which are occurring in the stroma. So some of the inputs and outputs, right, the reactants and substrates are familiar to us, right? We know that plants need sunlight. We know they need water. We know that oxygen is a waste product of photosynthesis probably. And we know that plants also need carbon dioxide and that we know that they produce glucose. So what we're going to look at today is the details of the roles that these different components play and how some of these waste products and some of these products are produced. But this is the overall. That the light-dependent reactions happen within the membrane of the thylakoid, whereas the Calvin cycle, those light-independent reactions are going to be happening within the stroma. And that we're playing now with these two important products, ATP and reduced NADP, also known as NADPH. Okay, so let's look first at these light-dependent reactions. So these light-dependent reactions, again, these are membrane-embedded complexes, and they are embedded in the membrane of the thylakoid. So one end of them is pointing to the exterior of the thylakoid, also known as the stroma, and then there's going to be some action going on within the thylakoid itself in the lumen. So what is happening in the light-dependent reactions is that light energy is coming into something called photosystem 2. And by the way, I just want to point this out. The chlorophyll, the pigment that is absorbing light energy, is located here in photosystem 2 and also here in photosystem 1. So those pigments that are absorbing the red and blue light, they're within these two important complexes, photosystem 2, and I know it's first and it's called 2, but that's probably the order that it was discovered in. The chlorophyll is located here and within photosystem 1 over here. So step 1 of the light-dependent reactions is that light energy is coming into photosystem 2, which contains the chlorophyll. And that light energy is being used to photolyse water. Okay, so photo means light, lysis means to break. So if I'm photolysing water, I'm using the energy from sunlight that was absorbed by the chlorophyll, and I'm using that energy to lyse water, to break it apart. And the reason for this is that this is an electron transport chain. Very similar, right, to what we saw in respiration, where I have complexes 1, 2, 3, 4, 5, and I'm using these reduction and oxidation reactions to create a proton gradient. I'm doing the same thing in photosynthesis. And just like in respiration, where I have 1, 2, 3, 4, 5 complexes, and that fifth complex is an ATPase, it's an ATP synthase, photosynthesis is also going to use a proton gradient. It's going to use chemiosmosis to also create ATP. So these are very related processes. They're very similar, right? But the source of those protons, the source of the electrons, is what is different here. So in respiration, right, we used our NADH and our FADH2. That's where our electrons and our protons were coming from in order to do the electron transport chain reactions. In the light-dependent reactions of photosynthesis, I still need electrons to drive this process. I still need to create a proton gradient to drive this process. Only now I'm taking both my protons right here and my electrons that I'm going to need to do this reduction reaction from water itself. So that's the first step, right? That light energy is coming in to photosystem 2, being absorbed by that chlorophyll, and I'm using that energy to photolyse water so that I can generate this proton gradient, right, that I'm going to need to power ATP synthase. And just like before, this proton gradient, this chemiosmosis, is being used to turn ATP synthase to create a ton of ATP. right? And just like the electron carriers were doing in respiration, I'm using this electron that was originally on this proton in water, and I'm using it to do a reduction reaction over here. And so we know that if I split apart water, okay, I take away the hydrogens, the only thing that's left is this oxygen here. So the waste product is going to be oxygen. The protons that were on water are used again to do chemiosmosis, which is going to power ATP synthase, the production of ATP. And the electrons that were originally on this hydrogen in water are going to be passed down through complex 1, 2, 3, 4, right? So into photosystem 1. And that electron is used to reduce NADP to NADPH. And so this guy is going to play into our next step, which is the Calvin cycle. So the two main products of the light-dependent reactions are the generation of a large amount of ATP from ATP synthase, which was allowed to occur through the generation of this proton gradient, and the reduction of NADP, from which I use the electron that was in the water to do this reduction reaction. And I'm also using the energy of light to complete this process in photosystem 1. So main points: photolysis of water with the energy of light to create a proton gradient that is going to then use chemiosmosis to make a large amount of ATP. Photolysis of water to also give me the electron and light energy here in photosystem 1 to reduce NADP to NADPH. So those are the main products of photosynthesis. And again, I'm just doing this to point out that there are pretty big similarities between these two different electron transport chains, that in each case I am doing some sort of either oxidation or reduction reaction. I am using a proton gradient in both of them to allow chemiosmosis to occur, that both of them are producing large amounts of ATP, and that both of them are within a membrane in an inner compartment of their respective organelles, the inner membrane for the mitochondria and the thicoid membrane for the chloroplast. And that both of them, again, are generating large amount of ATP from that proton gradient. Okay. So, light-dependent reactions were here, right? So this is the electron transport that's in photosynthesis. Our main products are ATP and reduced NADP. That would otherwise be known as NADPH, but for our purposes, IB is calling it NADP, so we're going to go ahead and use that language too. All right, main products, ATP and reduced NADP. So these products that were generated through these complexes in the thicoid membrane are then going to go out into the interior of the chloroplast, which is the stroma, and they're going to feed into something different called the Calvin cycle, or the light-independent reactions. And so what's going to happen in the Calvin cycle is that carbon dioxide from the air is going to be stitched together by our old buddy Rubisco. So this was one of the enzymes that we talked about in 2.4, and we know that enzymes either build things or break things. So Rubisco is using carbon dioxide to build glucose, right, which would be the whole point of photosynthesis. So this is a diagram of what the Calvin cycle looks like overall. So again, this occurs in the stroma. It occurs in sort of the open space within the chloroplast, and it's going to take in carbon dioxide, and it's going to also involve those products that we had from the electron transport chain. So somewhere in here, right here, is the reduced NADP, NADPH is going to be used to facilitate this cycle. And then you're going to notice that there's a lot of ATP that's going through this, right? About 9 molecules per cycle. So the energy that I generated, the ATP energy that I generated in photosynthesis, is going to be used within the Calvin cycle itself. Okay. So Calvin cycle has 3 main components, right? Fixation, where I'm taking carbon dioxide out of the air and putting it into a, physical non-gas chemical. So that's what it means to fix something, is that I'm taking a gas out of the air and I'm putting it into a chemical that is not in a gas form. So first stage is fixation, where Rubisco is taking carbon dioxide out of the air and combining it with something. The next stage is reduction, where I'm using that reduced NADP or NADPH to oxidize something else that's over here. And so we'll talk about that later. And that the last stage is the regeneration, right? Because this is a cycle. So whenever I use components, I'm going to regenerate something, this molecule right here called 3RUBP. So we'll talk about each of these steps in some detail next, right? So this molecule right here is called ribulose biphosphate. And it has 1, 2, 3, 4, 5 carbons within it. And it's called biphosphate because you have a phosphate here and another phosphate there, so that's 2. So it's ribulose biphosphate. A little sugar in here with 2 phosphates on the other side. But remember, our first step is fixation of carbon dioxide. And just a reminder that the enzyme that is doing that is Rubisco. So Rubisco is going to take carbon dioxide from the air and it's going to join it to this ribulose biphosphate. And so what it is going to create are 2 molecules of something called glycerate 3-phosphate. So this is a glycerate and it's got 1, 2, 3 carbons, and look, there's a phosphate on each one. And just a reminder that when I add a CO2 to something, right, that's a carboxylation. When I took a CO2 away, that was a decarboxylation. When I add one, that's a carboxylation. So Rubisco is combining CO2 with ribulose biphosphate. It's carboxylating this ribulose biphosphate to create 2 molecules of glycerate 3-phosphate. From here, a couple of things are going to happen. I'm going to use one of my energy bucks, ATP. And remember that I usually use ATP to take a phosphate group off and stick it on something else. So this molecule here, this glycerate 3-phosphate, is about to be phosphorylated using a phosphate from this ATP, and then something else is going to happen to these molecules. Reduced NADP, DP. Hello. So reduce, let's see where we are. Reduced NADP is then going to be used to convert these 2 molecules of glycerate 3-phosphate into triose phosphate. So I believe this is going to be oxidized, which means that the reduction is happening over here. So glycerate 3-phosphate is converted into something called triose phosphate. And so now I've got 2 phosphates on it where I only had 1 before. Just a reminder that one of those phosphates had to have come from ATP, so that's the donor of the phosphate. And then triose phosphate now has a couple of options. So triose phosphate can either become glucose, right? Because the whole point of photosynthesis is to make molecules of glucose. And if I have an excess amount of glucose, that glucose can then be stored in the form of starch, which in plants is either amylopectin, which is the branched one, or amylose, which is the spiraled one. So that is one possible fate of triose phosphate. But remember, we need to regenerate that ribulose biphosphate that started this whole cycle. So some of it is going to have to be regenerated into that ribulose biphosphate. And so this is the question, like if we have 1, 2, 3 carbons here, 4, 5, 6, and then this one is 1, 2, 3, 4, 5, how do I use